RNA, that is ribonucleic acid, its types and functions. See, all organisms, whether eukaryotes or prokaryotes, possess DNA as their genetic material. This biomolecule is double-stranded structure and it can undergo replication process to produce its own copies. From a small segment of DNA, the RNA molecule is synthesized, and the process of RNA synthesis from DNA is known as transcription. The RNA molecule could be of different types. They could be transfer RNA or messenger RNA or ribosomal RNA or a small cytoplasmic RNA. So I'm going to tell about the variety of RNA molecules which are synthesized and what are their individual functions. Now messenger RNA may undergo translation process, that is protein synthesis process, and by that process a specific protein is synthesized. So the flow of information from DNA to RNA and then to protein is known as central dogma of molecular biology. Now some of the RNA molecule may undergo reverse transcription to synthesize a segment of DNA. Now in this diagram you can see the double-stranded DNA structure, and the two strands are actually complementary copy of each other. One strand is in 5' to 3' direction and the other one is in 3' to 5' direction. It's one of the strand will be used as template strand to synthesize the RNA molecule. So in this lower strand you can see this one is RNA represented as 5' to 3'. Now this RNA molecule could be messenger RNA, or it could be transfer RNA, or it could be ribosomal RNA. But most of the RNA are actually synthesized quite long, but they undergo processing and then a mature specific kind of RNA is actually synthesized. So in this diagram you can see the flow of information from DNA to RNA. Now if you see the different types of RNA molecule, we first categorize this RNA into two major types. One, messenger RNA, which undergoes translation process to synthesize a specific protein. Actually on the messenger RNA, nucleotides form codon, and a specific codon means actually one codon is represented or it is formed by 3 nucleotides, and that encodes for an amino acid. So messenger RNA is used for mainly protein synthesis. And then the other variety of RNA molecules are non-coding RNA molecules. The non-coding RNA molecules are further classified as housekeeping non-coding RNA and regulatory non-coding RNA. Under housekeeping non-coding RNA, two major types of RNA molecule comes. One is transfer RNA, which actually recognizes a specific kind of amino acid and it brings that amino acid to the site of protein synthesis. Whereas the other one is ribosomal RNA. Actually there are variety of ribosomal RNA and they are actually required for the synthesis of ribosomes. So these are two housekeeping non-coding RNA molecules. The regulatory non-coding RNA molecules are of different varieties. They could be majorly classified as long non-coding RNA, which are actually more than 200 nucleotides, and the small non-coding RNA molecules, which are less than 200 bases or 200 nucleotides. The non-coding RNA molecules which are short, that is less than 200 bases, are again of different types like microRNA. They are just 19 to 22 nucleotides long. The other are small nucleolar RNA. They are actually required for the processing of ribosomal RNA and they are found in the nucleolus. Then a small nuclear RNA molecule. There could be small interfering RNA molecules. So these are small sized non-coding RNA molecules which I just said are less than 200 bases, but some of them, see, microRNA, they are just nearly 20 nucleotides long. Function wise, hnRNA, see, this is another type of RNA. Actually the synthesized RNA which is undergoing processing and which forms messenger RNA will be first referred as heterogeneous RNA molecule. So this heterogeneous RNA molecule are pre-mRNA molecule that serve as precursor for messenger RNA. It is synthesized in case of eukaryotes mainly in the nucleus. then it undergoes severe processing, like its introns are removed at its 5' end, a cap is added, then poly A tail will be added with this, and then it will be referred as messenger RNA. So hnRNA after processing is referred as messenger RNA, and this messenger RNA contains genetic information and therefore it is translated into protein. The third type of RNA shown here is transfer RNA. This helps in bringing amino acid to the site of protein synthesis. We know that there are several, amino acids, like 20 different amino acids are there. So to encode them or to recognize them, different kinds of transfer RNA molecules are required. Then ribosomal RNA, actually a precursor ribosomal RNA in eukaryotes is synthesized, that is 45S ribosomal RNA, but that is splits into small segments and forms 28S ribosomal RNA or 5.8S ribosomal RNA or 5S ribosomal RNA, which actually comes from a different part of the chromosome. And then the other one could be 18S ribosomal RNA. So such ribosomal RNA, they are actually required for the synthesis of ribosomes, and they provide a structure to the ribosomes, and ribosomes are required during protein synthesis. Then there are small nuclear RNA molecules which are involved in messenger RNA processing. So they are found in the nucleus. Then there are small nucleolar uh, RNA molecules. They are used for processing of ribosomal RNA. Then small cytoplasmic RNA are required. They are involved in selection of proteins for transport purpose. And guide RNA are also there, found in cytoplasm. They help in messenger RNA editing. Actually, some of the messenger RNA may require some kind of nucleotide addition or substitution or deletion. So for that purpose, a small guide RNA is required, which shows complementary pairing with the messenger RNA, and it will actually purposely change a specific nucleotide in the messenger RNA before it undergoes translation process.